Right. Good morning, everyone.
Good evening. Good evening.
Good evening. Good evening.
Okay.
Good
Hi, good
morning
morning.
to some of you. Good evening to some of you. Again, good evening. Good morning, everyone, and welcome to the webinar on global drug safety organized by the Association of Pharmaceutical Research, or the APR. Pharmaceutical sectors is booming tremendously, and pharmacovigilance is a major component. Drug regulation, clinical trials, drug innovation, and other health programs across the globe. Pharmaceutical professionals, scientists, and researchers are working on the drug safety monitoring despite of several challenges to make sure that we produce the safe drugs in the market. APR is organizing this webinar on pharmacovigilance to enlighten our fellow members on the challenges, recent advancement, and applications in the field of drug safety. Now, just a little bit of who our organizer, what is Association of Pharmaceutical Research. Here's a brief introduction about our organizer, Association of Pharmaceutical Research. Do you see my screen? Thank you so much for watching. And that's all about the Association of Pharmaceutical Research. To know more about our organizer, you can also check their website, www.assofarm.com. That's double S, ASSO farm.com. Next, I would like to invite our eminent speaker of the day. Firstly, I welcome Dr. Dinesh Jaiswal, pharmaceutical consultant,
Excuse
Hopesville,
me,
New York.
no, my timing is at about 5:40. I'm the third speaker.
Oh, I'm welcoming you. I'm welcoming you guys. Next, I welcome Dr. Dharmendra Kumar, Adjunct Professor of UCSI University, Kuala Lumpur, Malaysia, Professor and Head of Research at Laureate Institute of Pharmacy, HP, India. Also, Dr. Tosif Ahmed, the Vice President and Head, Biopharmaceutics, Preclinical and Clinical Bioanalytical, India. Thank you so much for imparting your valuable time in joining us today. Now let us welcome our first speaker. Dr. Dharmendra Kumar, Adjunct Professor of UCSI University, Kuala Lumpur, Malaysia. Professor and Head of Research at Laureate Institute of Pharmacy, HP, India. The topic is about the phytochemistry and phytopharmacology. Let me share the screen first.
Hello.
Yeah, but just
Hello.
just tell me if you want to go to the next page.
Okay, can I move this one? You can you
I
give me
can
command?
I can
You can
move
move.
it. Yeah.
Okay. So good morning and good afternoon to all of you, and good evening to all of you. Can you hear me?
Yeah, clearly.
Okay. So thank you, Miss Jojo, and all the eminent speaker, our Dinesh sir and Tausif sir, and all the participants. A very good afternoon and good evening to all of you. And I would like to thanks to organizer for inviting me as a resource person for this topic. This topic is very common topic, and it's a very general topic. It's the role of natural product in the drug discovery and development. It's a very general topic, very easy topic, but it's a very challenging for every scientist, challenging for every academician and researcher. So I am adjunct professor in UCSI University, Kuala Lumpur, Malaysia, and I am head of research at Laureate Institute of Pharmacy. It is situated at the Jalamukhi in Himachal Pradesh. And okay, so can you move to next slide, please? So our the introduction part, as I told, the natural product is nowadays is very important segment for the drug development and drug research, because in this pandemic situation, most of guys and girls are depending upon the herbal product or natural product. Even it's a very general example. Just I'm quoting you garlic or ginger. It's a very common product available in India as well as Southeast Asia and the different country. But the role of ginger and garlic, it has very wide role. So the natural products, including it's a plant, animals, minerals, and that based on the treatment for the human diseases. So about the if you think about the history of the medicine it has about the natural products. So majority of the natural products have been generated from the natural products, from the primary metabolites as well as secondary metabolites. But the main constituents from the secondary metabolites, from the compounds derived from the natural products. Even till date, most of the plants used as a biological activity or pharmaceutical activity, but most of the crude extract nowadays used for the activity. But till it has available the room for the many research, like you can go for the isolation, go for the characterization, go for the which type of compound it has biological activity. Like you know about the flavonoids. Flavonoids is a major component of the most of the plants. But the flavonoids, it has a antioxidant property. You know about the antioxidant because the oxidation. If you think about the oxidation itself, it causes a major problem, major health care issues, even diabetes, cancer, because of the oxidation, because that will generate the free radical formation. So these are the some outcomes of the natural products. Even in the 20th century, some of the pure extract of the plant, animals, and the microbes, even in the minerals, are represented only the medication available for the treatment for the human diseases, as well as for the domestic animal illness. So our new approach should be improve the drug discovery and drug research process and drug development. It's mainly that are innovative area. So you can think about it, and you can again move for the further process. So just I told you, because even ashwagandha, you know about the ashwagandha, it's a biological source of ashwagandha is a Videnia somnifera. And this ashwagandha, even in case of the pandemic situation, that many papers are quoted, ashwagandha act as a prevent the cell membrane entry. So basically, the major constituents of the ashwagandha is vithanolides and the vithanosides, and this vithanolides is it has a many applications. Now next slide, please. So our next that is called the natural products, because natural products it's a medicinal plant and it includes the microorganism. So there are some extraction methods, like you can say about the innovative extraction. So innovative extraction, it's subdivided into different parts, like the semi-bionic extraction, super SFC, you know about the SFC, super critical extraction, molecular distillations, membrane separations, and the ultrasonic assisted enzyme assisted extraction. So these are called the innovative extraction, even nowadays applicable in every labs. Now the second segment for the natural product for the extraction purpose is innovative. So innovative you can do for the automations. You can go for the CAT, that is called the computer aided drug design, and it includes QSAR, toxicity studies, and the combination therapy. In innovation, the next segment that is called the bioinformatics, and you can say about the multivariate analysis. And you are nowadays using the omics technology. The omics technology, it includes like the proteomics, genomics, metabolomics. These all are the some methods you can use nowadays for the extraction, for innovation, and for the further study. Okay, next slide, please. So the various process for the discovery of the drug from the natural product. So first point is the selection of the plant or the selection of the disease, the first point. So the first of all, you have to select the plant. Then after that, it may be the natural source, it may be the plant, it may be the mineral, it may be the sea origin, it may be the some animal source. So after selection of the plant, selection of the minerals, then you go for the primary screening. After primary screening, then you go for the phytochemical test. After you have to do the phytochemical test, then you go for the TLC, that is called the thin layer chromatographic studies. After thin layer chromatographic study, you can go for the column chromatography, HPLC, FTIR, GCMS. And after this screening, you can isolate the compound, you can characterize the compound of the active constituents. Because earlier I told you, you don't know the which type of constituents are mainly for the biological activity. So our aim should be recognize the active constituents, because which type of chemical constituents is responsible for, you say, like the anti-diabetic activity or anti-cancer activity. So after isolation, characterization, then you go for the structure elucidation because you don't know the structure. So after you collect the data from the different source like the GCMS, FTIR, OG, NOG, 13C-NMR, 1H-NMR, mass spectroscopy, and after that you can do the structure elucidation. Then after secondary screening and the primary screening, these are the interrelated. After that, you go for the preclinical development, and then I go for the tertiary screening. So next slide, please. Our next, that is called the some outline of the traditional bioactivity guided isolation. So you are facing, if you go for the isolation, if you go for the drug discovery from the natural product, you are facing many problems because these are the steps. Some steps are we have to select the organism. At the time of selection, the main issue is the problem number 1 and problem number 2. The problem 1 is it's not possible to culture the organism out of the natural habitat, and the second problem is the relevant natural products are not produced when the organism is taken out of the natural habitat. So how to solve this problem? You can solve by the new methods for culturing, and you can solve this problem with the help of new methods for in situ analysis. And for problem number 2, relevant natural products are not produced. Then on that time, you have to use the new methods for the natural products with the semi-synthetic source or the synthesis source in the induction of the heterologous expression of the biosynthetic genes. In problem number 3, 4, 5, that is called the presence of natural products that are not known. Suppose you have to extract, you have to isolate the compound, but after isolation, you don't know the presence of the natural products that are known. And even if you don't know the which type of drug-like property, if you can extract or you can isolate after isolation or after extraction, suppose the presence of natural products that do not have drug-like properties on that time, how to solve the problem? On that time, you can use the new method for de-replications, and you can use the new method for the extraction and the fractionation. So these are the some issues and how to solve these issues. So the things are very clear. The organism, the next step is the crude extract. After crude extract, then you go for the fractionation. And this fractionation or biofractionation, this fractionation will be repeated many times because you have to do multiple rounds of the fractionation. And it's a very major part of the any natural product. Fractionation, isolation, that is a major part of the natural product. And the second thing that is called the bioactivity evaluation. So at the time of fractionation, you must, I advise to all the researcher, all the academician, at the time of fractionation, you must do the bioactivity, in vitro bioactivity, because at the time of bioactivity, you can select the which type of compound or which type of fraction will be active. So fractionation is a very common and very important part, very important deciding part of the natural product. So with the help of fractionation, you can do the biological activity in vitro. And after which fraction will be important, which fraction will be maximum activity, then you go for the in vivo activity. You can go for the isolation. After that, you can isolate the compound and you go for the characterization of the compound. And after that, you can get the single component. So these are the some outline of the traditional bioactivity that is called the guided isolation steps in the natural products drug discovery. Now the next slide, please. So in the drug discovery process, it will take approximately 10 years, 10 to 11 years. And it's a very, earlier I told you, it's a very easy topic and very complex topic. It's very challenging. It's very challenging for every scientist. So for the drug discovery, the first point is the drug target selection is very important part. You can select the biological target. You can select the chemistry target. So you can select the enzyme target. You can select the disease target, disease as a target. So depending upon the first point is the most important point is the target selection. After target selection, it will take 0 to 2 years. After target selection, then you can go for the extraction, purification of the natural glycomolecules. Then you can go for the in vitro screening, approximately 200 to 500 samples. So it's a discovery and screening process. It will take approximately 2 to 3 years. Then after that, lead selection, and after lead selection, the time period it will take approximately 1 to 2 years. That is called, you can do the in vitro efficacy screening, identify the 10 to 20 best candidates. After that, lead optimizations, and the lead optimization will take 1 to 2 years. Then after that, marketing and post marketing surveillance, and then marketing surveillance. Next slide, please. So these are the processes of the drug discovery. So our the general plan is to choose a disease and choose a drug targets. Like target, it may be the enzymes, maybe the small molecules, maybe the different type of proteins. So first of all, you have to choose the disease, choose the drug targets, and the third stage, that is called the lead, find the lead compound. So after lead compound, that will be the optimize the lead compounds, and then you go for the clinical trial. So for new chemical entity, for NCE, it will take more than 10 years, and it's a very huge investment. And if you screen 10,000 tested compounds only, you can get only one compound as a drug. So these are the some challenging and very important information about the process for the drug discovery. Now next slide, please. So these are the some steps involved in the drug discovery, like the heat generation, lead generation. So H2L, that is called the heat to lead. It's a process. So earlier I told you the basic things are the lead identifications, target identifications, which type of disease and which type of target, that is important. Now the after that, then heat to lead, that is also called the lead generation, is a stage of the early drug discovery. So when where the small molecules heat from the high throughput screening, and it undergo the lead optimization. So these are all the process for the lead optimizations, and after that, and then you go for the candidate selection, and finally then the drug will be go for the clinical trial and the post marketing surveillance. So these are the process of the drug discovery. Now next slide, please. Now the next slide is, please. Okay, next slide. They already explained. So take now the choosing a disease. If you want to choose a disease, the pharmaceutical companies, the first aim is to make the profit for to exist, because every pharmaceutical company want to get maximum input, maximum output, and after that, the pharmaceutical companies will therefore avoid the products to a small market. So every company wants to go for the big market, because most of the common disease and most of the drugs are available in the market. So our pharmaceutical company, or the aim of the pharmaceutical company, to get the maximum benefits, and the drug will be widely distributed in the country. Okay, now next slide, please. So for the pharmaceutical companies, about the consider the economic factors. So as well as medical ones. So about the drug discovery process, the pharmaceutical company invested a huge amount. So it's a huge investment has to be made towards the research and development for the new drug. So therefore, the company must ensure the financial return. Obviously, it's a need for the pharmaceutical company. So the most of the research carried out nowadays, cancer, cardiovascular problem, metabolic disorders, depression, diabetes, migraine, obesity. These all are the major problem and major health issues of worldwide. And even nowadays, COVID itself a very major problem in front of all of us. Now next slide, please. So these are the drug discovery process. Clone the new targets, biological assay, screening, then lead compounds. These are the process of the drug discovery, drug development. About the pharmaceutical scale-up, then you can go for the ADME, absorption, distribution, metabolism, and excretion. That's called the pharmacokinetic profile. Then you can ensure the safety profile. Then after that, if the drug will be competent, then you go for the formulation, and after formulation, then you go for the clinical trial, and if the drug will pass the clinical trial, then you go for the FDA approval. So for the drug elucidation, you choose the model organism, you can choose the pharmacology, you can also fulfill the criteria of the genetics and the behaviors. Then after that, you can go for the new insight and targets and their mechanism. Now next slide, please. So identifying a drug target. So drug target, it may be the macromolecules, it may be the biological system, or it may be the any type of enzyme. So drug targets is the it may be the macromolecules that the drug will interact. And this drug identification of drug targets allow to researcher to identify the agonist as an agonist or antagonist should be designed for the particular receptors. Like one of the example, I know about the serotonin receptors, agonist of serotonin receptors, that is used the treatment for the migraine. But antagonist of the dopamine receptors are useful as these are the problems. So earlier I told you the drug targets is a very important part. And targets is maybe the protein, it may be the enzyme, it may be the anything, but you can select sometime agonist and sometime antagonist used as a different activity. Now the next slide, please. Now these are the drug targets. So drug targets may be water, maybe hydrogen ion, maybe metal ions, maybe enzymes like the digoxin. You know about the digoxin. Digoxin basically it's active constituents of digitalis purpurea, it's one of the plant. And this digoxin is a targets to sodium, potassium, ATPase. Nucleic acid as a target, and like the chemotherapeutics in cancer and the steroids. And the macromolecules, it may be the low specificity and the high specificity. So that's all are the drug targets. And before selecting the drug targets, you must be work on these all targets, which target is suitable for the activity and which target is suitable for the related disorders or diseases. Now next. So these are the some bioassay like the in vitro bioassay, in vivo, ex vivo. So in vitro, that is called the know about the in vitro. These are the definitions. So next slide. So these are the finding the leads. After lead findings, you can go for the natural screening natural products, existing product drugs, enhance the side effects, and starting from the natural ligands to the modulators and the combinatorial chemistry, CAD, and by chance, and the fragment-based discovery. Okay, next slide, please. So isolation and purification, as I told earlier at the time of beginning, because the lead compound is present in the mixture of the compounds, and you can obtain from the natural source. It may be the plant, it may be the mineral, it may be the animal source, and should be isolated and purified. So for the structural determination, you have to do the isolation part. After that, you can go for the characterization. You can get the data from the X-ray crystallography, mass spectroscopy, IR, NMR, mass, Koji, Noji, and after getting all the data from the different sources, then you can go for the structure determination, and you can say about go for the structural elucidations. And the next slide, please. So these are the processes. Now the drug development. So the process is bringing a new drug to the market. Once a lead compound has been identified for the process of the drug discovery, then you can go for the new drug development. So for the natural products, as you know, the richest source of the biologically active compounds, and many today's medicines are either obtained from directly from the natural source or the development from the lead compound that are originally obtained from the natural resources. Next slide, please. So the preclinical studies in the animals. So pharmacodynamic, all of you know about the pharmacodynamic, pharmacokinetic, toxicology, whether the drug is toxic or not toxic. So you can describe or you can give the single dose studies and the repeated dose studies. After single dose studies, you can get the check the acute toxicity, and after the repeated dose studies, you can get the data of the subacute, chronic, or long-term toxicity. And you know about the pharmacodynamic and the pharmacokinetic. In case of the pharmacokinetic, you are discussing about the absorption, distribution, metabolism, and excretion. So these are the some preclinical studies in animals. So these are done in the rodents and the non-rodents. And the clearance, these all process under the clearance of the IEC, that is called the Institutional Animal Ethical Committee required. So next slide, please. So the clinical testing, just I'm briefing the part. The phase one, it's you are dealing with the human pharmacology. Take the healthy volunteers, 20 to 50 subjects. In case of the phase second, you have discussed about the therapeutic explorations, and that's called the pertinent. The number of volunteers should be 50 to 400, In case of the phase third, that is called the therapeutic conformation, that is large scale up and the multi-center, and the number it should be 250 to 1000. And phase four, that is called the therapeutic use and the post-registration monitoring. Okay, next slide, please. So these are the some clinical trial processes. Natural products can be classified into the plant kingdom, microbial world. These are the some different sources. You can get the natural products from the different sources. It may be the animal source, it may be the microbial source, it may be the venoms, it may be the plant source, it might be the marine source. Now next slide, please. So just I'm quoting one of the example, like the morphine. Morphine you can get from the Purification, cocaine, digoxin, cocaine from the erythrogynon. I told about the digitalis or digoxin, digitalis purpurea. So these all are the some active constituents are responsible for the biological activity. So most of the biological active natural products are the plant secondary metabolites. It may be the alkaloids, it may be the flavonoids, maybe the glycosides, and that will be the responsible for biological activity, or that is responsible for the pharmacological activity. And the like quinine, suppose I am telling only about the quinine. Quinine is anti-malarial drug. You can get from the bark of cinchona, but this the dextroisomer of quinine, that is called the quinidine, and this quinidine is used as an anti-allergic drug. So sometime you can change the isomer and you can get the different type of activity. So this is about the some information about the from the natural source, from the plant source. Next slide, please. So these are the some structure. It contains the morphine from the paper somniferum. So these are the some derivatives of the morphine and the basic ring system. So okay, next slide, please. Now the quinine, you know about the quinine. Quinine you can get from the cinchona. So the basic ring system present in case of the it's a pyridine with the benzene. Benzene and the pyridine are fused together, and it's a cinchona. It's a plant that's plant's anti-malarial plant, and it's other activity as well. So the sixth position, it has the methoxy groups, and it's a quinoline ring system. Okay, next slide, please. Now the digoxin, already I told that digoxin and digitoxin, there is only one difference. You can get from the Digitalis purpurea, Splophoides family, and the wedge group and the non-wedge group is only the difference between the digoxin and digitoxins. Next slide, please. Okay, so the animal source, some of the animal source are very important for the biological activity, like epibetadine. This epibetadine basically is an analgesic activity, and you can obtain from the skin extract of the Ecuadorian poison frog. It's a pharmacologically active natural product. So sometime you can get the antibiotic peptides from the extract from the different type of frogs. So these are the some animal source, example of the animal source. Now the next one. Next slide, please. Next, that is called the epibetadine. This epibetadine is basically alkaloidal in nature, and earlier I told you can get from the frog. Then the next source as a natural products from the microorganism. So microorganism is another potent source of the drug leads, because based on these leads you can again can do the discovery. You can again check the biological activity, how much efficacy is, how much potential. So these are the some microorganisms you can get from the like different type of tetracyclines, like the chlorotetracycline, oxytetracycline, tetracycline, that are the drugs. And you can also get the penicillin, that is called the beta-lactam antibiotics. So these are the some examples you can get from the microorganism. Now next slide, please. And next, from the marine source. So marine sources like corals, sponges, and fish, and the marine microorganisms have very potent biological activity. It's very too much biological activity, like some of the marine sources used as anti-inflammatory, some of the marine sources as antiviral and the anti-cancer activity. So it contains some basic ring system, some important active constituents. That is one of them is called the curacin. Curacin basically it's we can get from the cyanobacterium, and basically it's a potent anti-tumor activity. This pyrazine is a proven activity for the anti-tumor, different type of anti-tumor activity. So the one of the important or richest source of the as a natural product as a drug discovery, that is called the marine source. And another anti-tumor agents like derived from the marine sources, including the these are the different parastatin and the dolastatin and the cephalostatin. These are the agents. But here, here the structure that's called the thiazole ring system present in case of the pyrazine. And pyrazine I can get from the marine sources. Now, next slide, please. So, as a natural product, venoms and toxins itself a biological activity and itself a source for the natural product. So, source as a natural product. So, venoms and toxins that you can get from the animals, you can get from the snake, spider, scorpions, insects, and it's a potent activity, and that activity, that constituents that will interact with the macromolecular targets in the body. So this venom you can get from the snake and spiders, scorpions. It's a biologically tools. It's act as anti-infective, act as insecticides, and you can take as a lead compounds. And that this all the active constituents you can get from the different microorganisms, different from different animals. Like you can also use as a drug targets identifications. So these are the some additional information about as a drug resources, as a drug sources. Now, next slide, please. So that is called another example, that is called the teplatides. This teplotide is basically peptide in nature, and this teplotide is a peptide you can isolate from the venom of the Brazilian vipers, and this was a lead compound for the development of the antihypertensive drug, because you know about the antihypertensive drug is the captopril is one of the famous drug. And this captopril is basically the basic ring system. If you see the structure is similar like the basic moiety as a captopril. So this teplotide you can isolate from the venom of the viper, and as a lead compound for the development as antihypertensive agents, captopril. So this is the agents. If you see the structure is similar. The basic ring system is similar in case of the captopril and the teplotide. So next slide, please. Okay, so conclusion. The conclusion is the natural products and derivatives has historically been invaluable as a source of therapeutic agents, and recent technologies advances to help address the issue, coupled with the unrealized expectation from the current lead generations, and approaches to have a renewed interest in the natural product as a drug discovery. Now, the various screening approaches are being developed to improve the easy with the natural products can be used in the drug discovery campaign and data mining as well as virtual screening techniques. These are all being applied to the database as a natural products, and it is a hope that more efficient and effective applications of the natural products to improve the drug discovery process. So these all are the some examples and the drug discovery process. Earlier I told it's very easy and very challenging topic to everyone, to every scientist facing the problem, because for the natural products, the main problem is yield problem. The yield is very less. If you go for the isolation, after isolation, the yield is very less yield. It's a major problem with the natural products. Second thing, that is called which type of compound is active for the biological activity. So there are many rooms are available for the further research. You can go for the molecular level study. You can go for the target level study, molecular mechanism. You can identify. After that, after you getting the result, then you the outcome should be future outcome for the society. It should be like in terms of the medicine, in terms of the patent, in terms of the data. That will be the helpful for the government, and many rooms are available for the research further in case of the natural product. Thank you so much for listening this topic. Okay, thank you, Ms. Jojo. Any question, please?
Anyone would like to ask a question? Right, seems none. Then let's proceed.
Because I have only 20 minutes. That's why I took 40 minutes. Okay. Sorry for delay. Sorry for I'm taking time because I only allocated only 20 minutes for me, but I took 40 minutes. Okay. Any questions, please? Audience, please. Any question?
All right, thank you, sir, for your valuable talk on the phytochemistry and phytopharmacology. Next, I would like to welcome our speaker, Dr. Tausif Ahmed, Vice President and Head, Biopharmaceutics and Preclinical and Clinical Bioanalytical India. The topic is about the bio waiver in clinical research.
Jojo, can you please project the slides?
Yep, I'm doing now. Sorry.
Thank you.
Okay.
Yeah, thanks a lot. So, am I audible to everyone?
Yeah, you are.
Okay. Thank you very much. At the outset, I would like to thank the organizers, APR, Ramya, Jojo, for inviting me for this talk. And also, it's great to be with sharing the podium with eminent speakers and a lot of audience. I see more than 50 audience. So we will get started. So basically, just to kind of give you some background about my talk, and I just saw a comment from one of the audience that they want something more on application based and more which can help on the employability. And I think my talk is exactly focused on that. So today we are discussing on drug safety clinical research. And if you see my top slide, I mean, what I've done, there's a human being and I kind of crossed it. The relevance of this is that we do a lot of clinical research, and I myself do a lot of clinical studies both in India, across the globe, in different countries. And we spend a lot of time, effort, money in developing those trials. And I think Dr. Dharmendra in his previous presentation showed that it takes more than 10 years to kind of bring a molecule into market, a new chemical entity into market, and primarily more than 50% of that time goes into the clinical trials. So one of the most updated area which is coming or which is upcoming is how we can avoid the bioequivalence studies primarily. It is not that you can totally avoid the clinical trials, because you have to do those for the new chemical entities because safety and efficacy is the prime concern. But there are when we are into I think from audience perspective, just want to give a perspective that there are two main streams into pharmaceuticals research. One is the drug discovery and development, where we work on new chemical entities. It could be synthetic or pharmaceutical or phytochemical, which Dr. Dharmendra referred to. What I am going to focus more is on the generic research. So generic research, basically in India, the drug discovery companies are limited, and somebody asked about employability. The options are limited in India, but in the generic space there are hundreds of companies, and very big companies, and I'm fortunate to be part of one of the biggest organization in India, which is Dr. Reddy's. And we do a lot of drug research in the area of generic drug development. With I will start with the disclaimer that the views expressed are mine and not of Dr. Reddy's. So I represent the Global Clinical Management Group at Dr. Reddy's, and I'm based at Hyderabad. Can we move to the next slide, please? Yes. So I'm going to cover some of the aspects of this during my talk. So what is the biopharmaceutical classification system? What exactly it means? How we can get bio waivers? What are bio waiver requirements? What are some of the ambiguities and some of the applications? And finally, conclusion. Next slide, please. Yes. So basically, as a very basic introduction, what is BCS? So BCS stands for Biopharmaceutical Classification System. It is a scientific tool or a model which enables the substitution or your replacement of the in vivo bioequivalent studies with the in vitro data. So just to give you a sense for those, I'm sure most of you are aware of what are bioequivalent studies, but just for the sake of everyone to be at the same page, basically when we have a new chemical, we have a drug which is approved. Take an example, very simple molecule, ibuprofen, and if we are trying to develop a generic version of that, which is a kind of a copy of that, what we have to do is we have to do a bioequivalence study. It is a study which is done in healthy human subjects, and you have to show that your product and the generic, the reference product, or which we call as RLD, are equivalent in terms of pharmacokinetic exposure, and then they are considered as therapeutic equivalent, and doctors can then substitute the reference product with a generic product. And obviously the advantage of a generic product is, and it is not that we are only developing for India, primarily we are developing for the regulated markets, mainly Europe and US. When we have a generic product which is of equivalent quality and therapeutic efficacy as that of the reference product or the RLD which we call, then it can be substituted, and the biggest advantage is the cost. So what I would see is there are two kind of so basically the drugs are classified into four biopharmaceutics classes, class 1, 2, 3, and 4. I will describe each in the next slide, but for a bio waiver where you can avoid doing these bioequivalent studies which I just explained, are two primary classes. One is the drugs which fall under class 1, and the drugs which fall under BCS class 3. And what are BCS class 1 drugs? Drugs which have high solubility, drugs which have high permeability, and in terms of dissolution, they're having either rapid or very rapid dissolution. Similarly, class 3 are drug products which are highly soluble. They have low permeability, but they have to exhibit rapid dissolution. I'm sure you are aware of the dissolution in those who are in the pharmacy field. This is one of the tool to assess the quality of the product in vitro, where you take the tablet or the capsule and do it in a dissolution apparatus, and then measure the amount of the drug which is released. And for class 3, it is also important that the drug should be Q1 Q2 same. So just to explain what is Q1 Q2, it is Q1 is qualitative composition and Q2 is quantitative composition. So you have to be similar to the reference product. Next slide, please. So since we are into a very regulatory driven industry, it is important to tell you what is the path of this bio waiver, or when we are talking of bio waiver of the bioequivalent studies, what is the regulatory pathway. So there are different regulatory guidances at different intervals which have come up from FDA as well as from the European agencies as well. If you see here, the journey of BioWaiver started in the year 1995, and it has now continued for more than 25 years. And as latest as May 2021, just a couple of months back, even then one guidance is released by the ICH, which is applicable to all the countries on the BioWaiver part. And I would strongly suggest the audience who are there, especially young students who are there, and academicians as well, who are interested in understanding what is BioWaiver. These guidances are available, and they are available in the FDA website. So you can go to fda.gov and search for these BioWaiver guidances, and you can get those guidances very easily. Next slide, please. Yeah, I think I'll cover this slide. Can you move to the next slide? I'll come back to this slide later. Yes. So as I was explaining, the concept of biopharmaceutics classification system, and the short form of which is BCS. So BCS basically means the drugs can be divided into 4 different classes: class 1, class 2, class 3, class 4, basis what? Permeability and their solubility. So if you know, in whenever you take any drug or any tablet, basically oral product I'm talking about, and these are not applicable to injectables, they're mainly applicable to oral products. So whenever you take a oral product, first it has to get disintegrated. If you talk about tablet, then it gets dissolved and then it gets absorbed. Basically, based on those physiological aspects, these the guidances have been developed. And class 1 are the compounds, and the examples are mentioned here, like nicotine, pregabalin, hydrocortisone, where they have high permeability and high solubility. Whereas class 2 products like posaconazole, which is antifungal, and the pioglitazone, rivaroxaban, which is anticoagulant, are compounds which have low solubility but high permeability. Similarly, the drug products like colchicine, lenalidomide, which is for oncology, they are the compounds which have low permeability but high solubility. So they will have permeability limited absorption in their mechanism. And mainly the oncology products will come in class 4, which have both low solubility and low permeability. So basically we are talking of biowaivers or avoidance of the bioequivalent studies for class 1 and class 3. For class 2 and class 4, they are not permitted, because you have to actually do the clinical studies to show bioequivalence with the reference product. Next slide, please. Yeah. So then since we talked of 3 main aspects of solubility, permeability, and dissolution, I will touch base each one of them, that what is the criteria, how they are done, what is the acceptance criteria, and how you can get the regulatory approvals based on this. And this is very industry oriented, and this is very application based. We use this approach to get the approvals of the generic products in all regulated markets. So first part is on the solubility part. So as per the guidance, both FDA and ICH, you have to determine the solubility in the pH range of 1 to 6.8, which is matching with the physiological pH. And a drug substance is considered highly soluble when the highest strength, that is, if you take a tablet of 200 milligram, then the highest strength is 200 milligram. It should be soluble in less than 250 ml of the aqueous media in the pH range of 1 to 6.8. And just to kind of bring more physiological relevance to it, as you see, whenever we take a tablet or a capsule, we take with a glass of water, and that is typically a volume of 250 ml. So if the tablet is completely dissolved in that, and this is primarily done with the API, then it is considered to be highly soluble. And then you can classify it in class 1, which is highly soluble. Next slide, please. So just to give you a concept of, see, in this 20-30 minutes, this topic is so vast, it is very difficult to cover, but my recommendation would be for you to go back and read these guidances. And in this short duration, we can only give you a kind of a introductory part of it. So when we talked about dissolution, we see that when we do dissolution or in vitro analysis before going into the clinical studies, we do dissolution as a quality control test. We also use dissolution to understand what is, when we develop different formulations, which is for formulation which is closest to the reference product before we go into the actual study. And we also do these dissolutions in physiological medias, which we call FESIV. Like if we are doing for fed state simulated intestinal fluid, you can do those dissolutions with this. And then the whole purpose of all this is that before you actually do your study in humans, you kind of go with the most closest formulation. Next slide, please. So again, on the permeability part, so we have talked about solubility, we have talked about dissolution, and what is permeability or what is needed in permeability. So for permeability, there are different methods by which you can determine, and we will go through them in the subsequent slide. So when we are developing a new chemical entity, one of the studies which are done are called as the mass balance or the MBS studies. Based on those data, if the bioavailability of the drug is more than 85%, it is considered as highly permeable. Or if there are certain in situ methods like intestinal perfusion methods, where you can determine the permeability, and the permeability should be more than 85% of the actual dose administered. Based on this, you can consider the product to be highly permeable, and you have to either use the literature data for this, or you have to generate this data to support your biowaiver application to the regulatory authorities like FDA, Europe, or WHO. Next slide, please. So again, for studying the permeability of the compounds, they can be classified into different categories. Like there are certain compounds which are low permeable or zero permeable. Like PEG is one compound which has no permeability. Then there are compounds which have low permeability, moderate, and high permeability. And when you do your experiment of permeability in a in vitro setup, you have to develop these methods in the you have to develop these methods in the experimental setup. Next slide, please. Yeah. So when you're doing these permeability studies, you also have to make sure that your product is stable and there is no degradation happening. So when you do these experiments, you have to prove that your drug product or the API which you are using, it should not degrade. So the degradation is cutoff is that it should not degrade more than 5% when you're doing these studies. So this you should keep in mind. And the experimental condition is that the incubation should happen at 37 degrees for 1 hour in both gastric fluids and 3 hours in intestinal fluid. And there should not be any degradation more than 5%. It should be less than 5%. And this is again to kind of mimic the actual physiological condition. Next slide, please. Yeah. So coming back on the dissolution. So what are the dissolution? If many of you, if you are in B Pharm or M Pharm or in your other academic institutes, you will typically do these dissolutions in different dissolution apparatus, which is USP1, the basket, or USP2 paddle, using different volumes. And you have to generate the dissolution data in the different conditions, similar to what when you administer a tablet, it will first undergo in acidic, then it will be pH 4.5 and 6.8, and you have to do minimal 12 units of tablets or capsules to generate this data. And for comparison, you have to use a statistical test to show similarity between the test and the reference product, and it should be more than 50. The value should be more than 50, then it is considered similar. Next slide, please. Yeah. So as I said, for a BCS class 1 product, the drug substance has to be highly soluble, highly permeable, and rapidly dissolving. And again, we will come to the excipients, but one of the important part in any formulation is the excipient. So the excipient also should not impact the rate and extent of absorption or the pharmacokinetics. And for class 3, it has to be highly soluble, very rapidly dissolving. So very rapidly dissolving also, we'll see what is that number. And it has to be qualitatively and quantitatively similar. If you recall, I mentioned terms Q1 and Q2. This is where qualitatively is Q1 and quantitatively is Q2 in terms of formulation and excipients in the formulation or the test formulation or the generic formulation which we are developing. Next slide, please. Yeah, so additional requirement which we see is in terms of excipients is that we should not have any excipient which should impact the bioavailability, and it is more stringent for BCS class 3 compounds because they have a permeability limited absorption, and the composition of the test product must be quantitatively the same and quantitatively very similar to the reference product. Next slide, please. And there are also certain cases or classes of drugs where this waiver cannot be applied, and those are like narrow therapeutic index drugs like phenytoin or diazepam, where the safety is a major concern, where the biowaiver is not applicable. And then the products which are kept in the oral cavity, like they're not ingested or taken orally, but they dissolve in the oral cavity. And also another category of drug products like orally disintegrating. ODT stands for orally disintegrating tablets, which are kept in the mouth. The bio-waiver is also not applicable to this. And to reiterate the fact, the bio-waiver for based on BCS classification is only applicable to bioequivalent studies. They are not applicable to phase 3, phase 4 clinical trials, or they are not applicable to the food effect studies. Next slide, please. Yeah. So again, when we are generating the data on the solubility, the methods or analytical methods which we use, they have to be fully validated, and we have to generate both the mean, standard deviation, percent CV, and a graphical representation of the pH and solubility profile also needs to be submitted. So this is our practical experience. If we don't submit this data, we will get regulatory deficiencies and we don't get approval, and we are asked to do the bioequivalent study, which is again very cost intensive and time intensive. So the data generation is very critical in whatever area you are. Next slide, please. For the data on the permeability, the methods which we use, they have to be validated, and you have to whatever tool you are using, if you are generating the data in the in situ or ex vivo permeability assays, you have to validate those with model compounds, as I mentioned, as low permeable, moderately permeable, and highly permeable class of compounds. And you have to generate the plots with almost 25 model drugs as a supportive data, that the methodology which you're using for permeability determination is robust and valid. Next slide, please. Similarly, on the dissolution part also, you have to use equipments which are calibrated. The methods which you use and the buffers which you use are also of high quality, and you have to provide the description of the method. You have to provide the description of the lot numbers, and you have to do statistical and analysis, the graphical representation of the data. Sorry.
Oops. Are you guys, sir?
Sorry.
Go ahead.
Yeah, sorry, there was some issue in the so again, coming back on the excipients and the manufacturing process which we use, the manufacturing process for the methodology, whether it is wet granulation or direct compression, has to be described. And excipients is also very important, as I explained in the previous slide. What amount you are using, what is the comparison, what is the intended function, that has to be provided. And you have to have proper communication and correspondence with the regulatory agencies for getting the approval. And Q1 Q2 sameness has to be proven, especially for class 3 compounds. Next slide, please. Yeah, so again, I don't want to go through this. You may get a copy of this, but what I just want to show here is that there are certain differences in the requirements between different regulatory bodies. Like if you consider FDA, which is the US regulator, and if you compare with other markets like Europe and Australia, Canada, there are certain differences. I will just highlight some of the differences about their requirement. Like if you see solubility, FDA asks for the highest strength, whereas Europe asks for the highest dose. Like if you have a 200 milligram tablet, but the dose is 800 milligram, you have to generate the solubility for US for 200 milligram, but for Europe, you have to generate at 800 milligram. Other requirements are more or less similar, but there are some minor differences in terms of time points or in terms of specific requirements. Next slide, please. Yeah, similarly, in terms of excipients, also FDA is giving more clarity in terms of prodrugs or what are the exclusions or which where biowaiver cannot be given. As I mentioned, oral cavity, buccal sublingual tablets, and narrow therapeutic index cannot be given as per FDA. Whereas for EMA, the esters, salts, or buccal sublingual cannot be asked for biowaiver. Whereas in other cases, it is more or less similar. Next slide, please. Yeah, I think we are just coming to the last few slides. So basically what we have done or what we have seen is one of the approaches for avoidance of the unnecessary human clinical research, and BCS based biowaiver is one approach. And this is taken from a regulatory communication that the focus is to prevent the unnecessary use of humans in the clinical trials for ethical purposes, and also to ensure the quality of the products and right time first, and also to ensure that time and cost of development is reduced. Next slide, please. Jojo, can you change the slide? Yes, thank you. So this is the last slide. So essentially what message I wanted to give is that this is a very novel, very recent concept where you can get the avoidance of the biowaivers. You can get biowaivers in avoidance of clinical studies for class 1, class 3 drugs, and generic companies are making effort on this line. And you can also make an effort to avoid some of the patient pharmacokinetic studies and can avoid time and effort. And recently ICH M9 guidance has come, which I would refer that you people should refer to get more understanding. And in future also this area will keep on evolving, and to investigate the possible extensions of the current biowaiver, we can also use approaches like mathematical modeling and simulations such as PBPK modeling, which is also something which is totally new and upcoming. With this, I would like to end my presentation. I want to thank the organizers, and if there are any questions, I'm more than happy to answer.
Any questions? All right, seems none. And thank you so much, sir, for your amazing session.
I think somebody has mentioned, can you repeat the modeling which you were telling? Yes. So basically the modeling concept is the physiologically based pharmacokinetic modeling, where you can use the in vitro data and the available literature data, and you can use that data to prove that your product is similar and efficacious. No, and it is not admit. It is not admit. It is basically the dissolution data and the PK data, human PK data of a similar product available, where you can use that to develop the model and show efficacy and safety for your product. So maybe you can do some more literature search and you'll get more idea with that. Thank you.
Thank you so much. And before we proceed, and just check my recording. All right, let's go ahead and proceed. Now, I would like to welcome our next speaker, Dr. Dinesh Jaswal, pharmaceutical consultant, Holtzville, New York. The topic about the clinical trial materials. Sir?
I would like to share my screen,
Yep, go ahead, sir.
but it mentioned the host disabled participant screen sharing.
Oh, my apologies. One moment.
Can I share now?
Yes.
Now it is visible now?
Yes, it is.
Hello?
Please. Yes, it is. Please proceed.
Okay, just now we heard the good morning and good evening to you all. Just now we heard about a very good speech from Dr. Dharmender and Dr. Ahmad. Dr. Dharmender gave a very nice speech regarding natural product, and Dr. Ahmad gave you some feedback regarding industry, the generic products. And I would like to tell you during this COVID crisis, at least we pharmacists are well organized, recognized. Yes, I'm not telling the doctors are very good for the diagnosis of the disease, but we pharmacists are not going to produce any medicine what doctor will do. That's why we are very playing a very vital role. And pharmacy is a very highly uh, noble and honest what you call profession. And today's my topic is release process of clinical trial materials. Before that, I would like to tell you something regarding difference between the brand name and generic. And people are always complaining that why brand name is so expensive. Let me tell you in short over here, because time is very less. The brand name, because a company is working for 10 or to 15 years and at 10 or 15 products, and they got one product approval after 10 or 15 or 5 years for the into product approval from the FDA or any other agencies. And in that process, when a company wants to take the prices of the other drugs, which they are already uh, spent on that one, that's why if they got one product and they got the patent for the product for 5 years or 10 years, it depends on the product. And that's why the brand name is so expensive. And for that one, we have to make a lot of clinical trial materials. That's why in the different phases and all those things in different country. And I will give you some small feedback regarding the clinical trial materials and what are the release process. And these are the outline of my presentations. I will give you some background and then some key components and role of team leader. And that is my main presentation is focusing on those who are interested to join the industry, pharmaceutical industry. And sometimes you have to site visits and meetings. And sometimes in the company, if you work over there, there's a lot of differences and overcoming, because the pharmaceutical industry have a lot of departments, and which I will tell you the later part of my presentations. Then we have to do a teamwork, we have together everyone achieve more, we have to do the teamwork. And then after that, I will share some examples of clinical trial materials, global stability, packaging issues, and finally, I will conclude. Suppose in the manufacturing, you have to make sure that there should be less problem in manufacturing. And suppose any time the day changes, we have to be very sure that you have to make the implementation of new technology. And documentation plays a very vital role in the pharmaceutical. Yes. Any problem? Hello? Hello, any problem?
No problem, sir. Go ahead. Oh,
And then documentation plays a very vital role. And so that you have to be, I have to talk something about the documentation. And everybody should know in the pharmaceutical industry or pharmaceutical student, the current good manufacturing practice, the routine follow-up, and then coordination with various departments, which I discuss later part of my presentations. And these are the four key components you have to always remember, that you have to communicate very well. Suppose sometimes in the industry, if you are not very good in communications, suppose your speech and all those things is a different action, then you have to write the emails and all those things, the written documents. The communication plays a very vital role. Then we have to plan very well. And at the same time, we have to be teaching ability for the demonstrations. And as I mentioned, the documentation is also very important. Then team leaders. Suppose in any projects, all the team leaders should be there, and different project team leaders are selected. And the major team leaders must ensure that everyone on the team understand their role, get done in a very timely manner, or all the projects should be done in a very timely manner. And the team leaders should be very well organized, understanding the process involved, and must motivate the team, because they should not, every human being has got some positive points. They try to motivate and provide positive influences. Don't talk about anything negative over there, and task functions highlighting priorities. And then you have to maintain the status report of each product, and then encourage team members and manage their activities. And we need to the coach whenever it is needed. And if you go to an industry, some people are the troublemakers, but you should not talk about the troublemakers in front of everybody. You should talk separately. If needed, then you have to talk to the director or vice president of the company. And suppose somebody still is making trouble, then we have to show them the rule, show them the door. And we have to always resolve conflict also. And sometimes the pharmaceutical company has not got all the facilities. Then you have to go for some other facilities, and then you should know to site visits while products being manufactured and packaged and tested in a transfer site. Because sometimes you have to, the company, they don't have all the facilities, they have to go to the other sites also. And while you transfer, it is not a paper transfer. You have to make all this product and everything. You should be know to go to over there and to find it out what kind of equipments they have. And at the same time, you have to gain experience to visit the transfer site, worth of time and effort with the products and methods from the associates that know the product best. And you always make an agenda, should be issued well in advance, because not at the same time, all the agenda should be well in advance, allow the team to stay focused and helpful in time management, and very useful to have regular meetings. Regular meetings, we should know everybody, because in the pharmaceutical industry, so many departments are there. You have to make a regular meeting when team member can report on the progress of each activity and task. Because it's not a very simple project. We have to do a lot of research for the 10 or 15 years. We always to maintain the progress. And minutes of the meeting should be issued within the same day or must be within a week. And minutes and agendas should also be sent to the top management of the related departments. And sometimes we are working, they will get some differences in the society and sometimes difference in the company. Difference in the society, suppose sometimes any product, suppose in India, the culture is entirely different in the south and as well as in the north. So you should respect the culture, you have to respect the language, philosophy, structure and regulation, budget and market activity. And suppose, as I mentioned it, you have to learn the culture. If you learn the culture and you have to respect the culture, and sometimes if you're transferring from product from south to north, then you have to know some local phrases so that you have to work with them, with their employee, and meet your counterparts in person and build trust on them so that we can very friendly atmosphere, they can trust to all of them. And understand the organizational structure with that company. We have to understand what kind of organizational structures is there. Bottom line, have an open mind and do not be afraid to use common sense. And in the company, sometimes, as I mentioned very clearly, all the pharmaceutical company has got different departments. Sometimes we have to be scientific, then discovering, learning, and tight controls and marketing demand. Regarding marketing, I will tell you the later part of my presentations. And the company, the quality control and assurance should be there. And sometimes quality control and assurance, we cannot fight with them. We cannot force them. We have to make a very friendly atmosphere with them. Compliance, regulatory, purchasing, manufacturing, analytical, etc. And sometimes you have to utilize a cross-function team and educate other team members. You have to educate it because some department, they don't know what is their function, but sometimes being a leader, you have to educate and prepare a project plan together. Marketing approach. Marketing approach, let me tell you, in the pharmaceutical industry, marketing is very, very powerful. If suppose you make a product and suppose in the round bottle, if marketing will tell at the 11th hour they want to sell the product in the square bottle, that's why in very early stage you always involve marketing people. What people are doing, if they have any objection, they can tell you up front, not at the later part of the work. And development approach, operation approach, timelines, and any other related issues. And some people think in the company they are working only one person. Not. Any product is a group of work. So some person, they think the department develops a product, no one person. Successful teamwork depends on a strong leadership, clear role and responsibility, skill, technical organizations, and communication, as I mentioned earlier, and problem solving, and you have to build a trust. And that's, you have to be very professional. We have to maintain the human relation and resourcefulness. Suppose sometimes any product, some information you need, you have to find it out from where you can get all this information. And you have to also learn some management knowledge and analytical approach to any problem solved. Every time any kind of problem will come, we have to be ready to solve the problem, and you have to have some confidence and youthfulness. Always you have to approach to a problem like an energetic youth. And also you have to very clear project objectives, strong leadership, minimization of defensive attitude. Some people in the industry, they always work, I did not do, they always try to be defensive, but it should be, if any problem comes, you have to tell them that this is the mistake we have done, that we have to solve the problem. Don't apply any defensive attitude. And appreciation of culture difference, so you have to appreciate. Keeping ego. You should not be very proud and something that always keep your ego outside the door. And self-awareness, responsibility, members with motivations and support. Of course, we need a top support from the top management. And one-to-one discussions and celebrate completions of any key activity. Suppose any small activity is completed, we have to celebrate, and you have to recognize all the employees for a job well done and show respect no matter what level. Even a technician, even what kind of level, you have to show respect and ensure good team member, team leader relationship. And dedicated members cannot, suppose sometimes a dedicated member, you don't need a member. You always say yes and yes. You have to, for some person, they should give a concrete example so they can follow. Never ever expect any yes person every time. And members cannot just give completion dates. Dates must be realistic and doable. Now I will tell you the clinical trial materials release process and release of all clinical batches without QA observation and in a timely manner. Though you have to always make sure that all the finished product and all those things should be released by QA. You have to make always very good relationship and always update QA. And we have to convince them whatever. Suppose sometimes QA person, they are not very highly technical person. We have to convince them so that your materials can be released on a very timely manner. And QA, they always give the documents and all those things. As I mentioned earlier also, the documents should be well organized and immediately available for any audit. Suppose any FDA comes or any agency comes, your documentation system should be in such a way so that if any inspector wants this kind of documents, we have to give them within a couple of minutes or 5 minutes. The moment we'll more a delay, then they will think that there's something is a problem on that one. If we'll give the documents well in time, they are not going to ask any further questions because they knew very well that all the documentation are very crisp and clear. You have to plan, organize, leading, and controlling. Make decisions based on scientifically justifiable facts, data, and trends. To guarantee product quality and meet positive business results, need to plan proactively. Always you have to plan very proactively approach. And every pharmaceutical industry, all the persons should be find it out, should be very proactive and make clear and effective decisions and coaching. Maintain always motivated environment. The review of manufacturing product protocol. Any batch you have to prepare, you have to pick the manufacturing protocol, and then after executed batch record, batch dispense record, all those things. And review of excipients, release data, manufacturer certificate of analysis, drug substance release data. Suppose if you are buying any excipients or API, the certificate of analysis is given by the company, but at the same time, all the excipients and APIs should be tested in the company so that there should not be much difference between certificate of analysis plus minus 2% is okay. But we have to maintain the same. We have to compare this comparison and review of analytical data, review of product certificate of analysis. And follow up with the formulation, because as I mentioned very clearly, in the pharmaceutical industry, so many groups are there, so many departments are there. We have to follow up with a formulation group, tech transfer group, quality control, manufacturing, analytical and clinical packaging on routine basis in order to release all supplies in a very timely manner. Suppose sometimes the timely manner that I'm telling, suppose the marketing people and medical group, they created some patients to enroll. So your material should be released very in a timely manner. Otherwise, the company will lose millions of dollars, something like that. And create product release checklist and coordinate with QA for final release as mentioned clearly, and organize all the batch related information and folder that could be used for future reference. You have to maintain the informations all in a folder, which I will tell you the later part of my presentation. And so clinical trial materials should be released in a very timely manner. I just give an example. It depends on the company. The first stage should be the manufacturing process. The second stage, analytical testing and release of the batch stage. But it depends on the company requirements. But as based on my experience, I push it there for 15 days. You have to issue a master batch record and batch number. It depends on the company. And we have to prepare the manufacturing protocol, approved manufacturing protocol. Then after the manufacturing protocol, we cannot make any changes during the production. And the manufacturing process and preparation and approval of the specifications if needed. And then sampling of API, as I mentioned, that active pharmaceutical ingredient excipients to the QA, to the analytical laboratory. And then release of API and excipient for first 15 days they can do in the lab. And review of internal certificate of analysis, I mentioned, and vendor certificate of analysis. So there should not be much difference on the whatever internal certificate of analysis and vendor certificate should be matched. Plus minus 2% is okay. And review of the executed master batch record. When the batch has got manufactured, the master batch record can be reviewed and submit the executed master batch record to QA. And then submit the samples of finished product for analysis. Finished product sent to the clinical packaging at that time after, and the review of the executed master batch record by QA so that they can check in that period. And analytical testing of the finished product, and the finished product analytical testing can be performed, and preparation of analytical release documents and audit of the analytical data by QA. They can do audit whatever the QA, and sometimes the QA have got the audit of the analytical data and audit of the formulations group, the different QA, but depends on the company. And preparation and approval stability protocol. At the same time, you have to prepare the stability protocol also. And finally, the certificate of analysis to be issued for finished product, and review of the documents, approval process of the document And at the same time, you have to make the stability setup, which I will talk to you later part. And suppose in the folder, we have to keep all those things. Suppose after 4 or 5 years, FDA comes, so what kind of product, some confusion, you have to make the folder for the safety. The products in the folder, you can put product certificate of analysis, manufacturing protocol, batch record informations, drug product specifications, drug substance specification test method, drug substance release data, excipients release data, and manufacturer certificate of analysis. And product certificate of analysis and manufacturing protocol. And in manufacturing protocol, it should be detailed sampling, manufacturing plan, and testing of the drug product in accordance with the master batch record. And stability data also includes the clinical stability data gathered and tabulated in the durations of the study. And for information purpose, if you need anything, all documents that is not pertinent to the releasing process, but you can keep in the folder also, clinical batch, however, all included for the documentation purpose for future review. Let me tell you the packaging process. Because some company, they cannot afford all the kind of equipments and all those things. The objectives should be there. So whatever based on the company availability of the instruments and all those things, you have to make the successful packaging process from the very early development stage to market the product. Promote effective communications between R&D, commercial packaging, and related departments, namely marketing and sales. Provide clear and concise directions to obtain a positive results, and work effectively as a group with various departments to meet corporate goals. That is always objective. We have to meet the corporate goals also. Considering clear concept and the product integrity, the R&D packaging development group will generate the stability data based on the various experimental packaging design. R&D research and development will recommend selected container closure system to commercial packaging group after careful evaluations of the stability data. And R&D will also provide a placebo. We have to make a placebo also so that the commercial packaging group, they can maintain what kind of equipments they need, whether it is available or not, along with the theoretical running weight, the tablet to determine the fill volume, etc. The commercial packaging group will determine the fill volume of the container closure system based on the available inventory, machinery, and equipments, etc. If that's not available, then you have to go for the outside of the company. And after approval from the commercial packaging group, the packaging request order will be initiated by R&D. And see, I will show you some example of the packaging request order form, that you have to make the product and you have to description of the bottle very clearly, description of the cap. We are putting a desiccant, yes or no, and bottle fill count and other things requirements. It depends on the company. We can prepare of your own. Based upon the approval packaging information, the packaging operations will be coordinated by the commercial packaging group or by outsourcing, as I mentioned earlier. The R&D group will be responsible for releasing all the related container closure system to satisfy the NDA requirements, because that is R&D responsibility for the new product. It's not a marketed product. When the marketed product will be there, when it got approved, then R&D is not responsible. But in each case, R&D is responsible. Any problem comes, R&D has to solve this problem. And R&D will be responsible to collect the certificate of analysis along with the vendor certificate of compliance and all related documents, namely related diagrams also. And these are another examples, the packaging parameters for bottles, description, manufacturer, size, shape, resin, colorant. You have to put all this information, what kind of resin, what kind of colorant. And for caps also, who is the manufacturer, size, inner liner, and induction shield if it's there. And for the rayon, suppose if there, then for blisters, we have to give all the information regarding film. It is a monolaminate, bilaminate, or trilaminate. It depends on the product, what we're doing the stability. And the packaging authorization, then we have to take the authorization from the R&D, definitely, commercial packaging group, marketing, and sales. We get the approval from them, then we can start the process. This is the global stability program. It depends, and I will tell you in a very short because time is very less. And long-term, intermediate, and accelerated condition, these are the conditions. Suppose for the clinical, at least you have 12 months data is available. Suppose 25-60 is not desired for some climate zone, then you have to go for the intermediate, that is 6 months. Then accelerated condition 40 and 75. It depends on that one, the product. And these are the lists for all the countries I mentioned, and you find what kind of atmosphere we need and what kind of temperature we need for that one. And there's 4 climatic zones. It is mentioned it will go 1, 2, 3, 4A, and 4B. And it depends on what kind of dispersion and temperatures, subtropical, hot, dry, hot, humid, very humid, and what kind of temperature you need, and accepted long range stability data. A reduced design, sometimes it is not possible to do all the stability tests. Suppose there's so many different, so we can do what you'll call the bracketing and matricing design for that one, but based on the formal FDA get the approval. Suppose a product is a dose proportional and the strength is about 1.5 milligram and 12 milligram. We need not to test all the test station. For 1.5 milligram lowest strength and highest strength, we have to do all the testing, all the states for 3 months, 6 months, 9 months, whatever. For the batch 1, batch 2, batch 3. But other strain in between, we can reduce some testing, but we take the approval from the FDA. So in order to reduce the testing, for this is good for the dose proportional product. And this is another examples for that one, so that in order to avoid some testing, you can make these parameters for matricing design. And this is the protocol setup, sample pull, and chamber monitoring. We have to check the chamber. Suppose chamber is 25-60, it is monitoring. Sometimes it is due to light fall, and sometimes the temperature is not there. We have to maintain every day. And suppose sometimes the temperature reflects, then we have to take the samples out, and we have to test for that one. If suppose due to some, then we have to make sure you send the samples to the other outside the facility. We have to make the protocols well in advance and SOP for that one. And the execution of all ND and clinical support protocols, review protocols to evaluate and allocate samples needed for the duration of the study. What kind of samples, for how many samples they need, we have to do it well in advance. Preparation and setup activities, generate sample bin labels for stability setup. That is also very important factor, and stability group, they have to be very careful. They are monitoring. They should not contaminate with other products. That's the bin levels and everything well labeled on that one. And FDA comes, they always go to the stability area and chambers also. They have to be very careful on that one and be very positive. Each sample is labeled with sample levels. Time zero determination, submit time zero as needed. Suppose you got a manufacturer, generally manufacturing date when you API mix that one. And for the time zeros and everything is done within a month, we set up the stability, and from the time zero start from that period. And generate pull list and labels also. So well in advance is schedule samples and pull. Each sample is labeled with the label containing sample ID, and requested samples are submitted to the lab. And sometimes we have to put additional samples from the lab. It could not able to, and there may be some destroy. Then we have to give the optional samples are submitted upon request. And suppose, as I mentioned, is a pharmaceutical industry. You cannot destroy the samples as these. We have to, stability inventory management should be there. Identify sample for destruction based on completion or termination of the stability. We have to accountable for all the samples, how many samples got destroyed, how many samples got for testing. And let me show you some stability chamber chart. So we have to maintain the chart on regular basis. It is 25 or 40 degrees there. We have to maintain that one. The FDA wants to see the chart. So we have to maintain the everyday charts like that. These are the typical examples. Now, let me conclude. In order to ensure the successful launch of the new pharmaceutical product, it is imperative that a complete necessary technology be accomplished. Rational and scientific supports for the specification and process that are established for each product should be available. FDA and any agency expects that the company will have developmental data and all reports to support all relevant process and testing methods used during the course of a development and transfer of a projects. FDA especially interested reviewing of supporting data and informations about changes that have been made at each phase of the transitions from R&D to commercial scale. For example, establishing and reviewing specification, scale of batch sizes, modifications to the method, manufacturers, and composition of the finished product. And mutual understanding and strong communications between the departments involved is essential. Flexibility on each side is also crucial. Sometimes you need to make changes, but when adjustments are necessary, we have to be flexible also. But we have to inform to the FDA any changes and all those things. Developing a process for commercialization of the product into the market is important, and positive communications amongst the group is the key for a successful process. Thanks to everybody.
Thanks so much. Anyone would like to ask a question? Anyone would like to ask a question? All right, seems none. Again, I would like to thank Dr. Dinesh Jofwal for your very informative session on clinical trial materials. Now, I personally acknowledge and appreciate all of you guys for sharing with us today, in spite of this time difference, in spite of the time difference, the good morning and the good evening. With this, we come to the end of our short webinar on global drug safety. I would like to thank all our three amazing speakers, Dr. Dinesh, Dr. Tosif, and Dr. Dharmendra, for imparting your knowledge on pharmacovigilance. I would also like to thank all our APR members and YouTube viewers for your patient listening. Last but not the least, I would like to thank our scientific partner, BioLeagues Worldwide, and research partner, United Innovators. Thank you all once again. Moderator Jojo, have a great day. Good night, everyone.